So,
I don't need much introduction.
no, it's good.
You've said enough already.
I've said enough. So we had a rather non-molecular discussion at breakfast, and so the contrast may be a little too great. I apologize in advance. But I'm going to talk today about nicotine addiction from the point of view really of the nicotinic receptor, and we're going to go much more toward the biochemistry and the molecular biology of the receptor. So I hope that there won't be a big overlap between what you heard from Paul Kenny, because he told me that he spoke a lot more about the systems level, and this is going to be much more about the molecular level. So stop me if there's something that you really don't want to hear about, and do stop me with any questions going through the talk. I am happy to take this wherever you need it to go, and we'll go from there. So here is the molecule we're going to be talking about, nicotine. Small, charged, basic. And one thing that I want to start with is the fact that although I'm going to talk about nicotine pretty much exclusively, it is one of 4,000 constituents in tobacco smoke. So we know if you take nicotine out of tobacco, that people will extinguish. The majority of smokers will actually, over time, extinguish, probably because they have choices of other tobacco products with nicotine in them. But there are other compounds in those, among those 4,000, the Harmala alkaloids. There are molecules that are GABA-A agonists, so they will actually act like benzodiazepines. There are molecules that are MAO inhibitors, so they will increase dopamine on their own. And there have been a number of studies now trying to combine nicotine with these other types of, let's say, classes of molecules, because the actual molecules in tobacco smoke have not been used in large quantities in most of the basic science experiments. But you can get synergistic effects, or at least additive effects, of some of these other constituents in tobacco with nicotine. And I want you to keep that in mind, even though my laser-like focus on nicotinic receptors will be the rest of the talk. So we didn't obviously evolve nicotinic acetylcholine receptors in order to roll up the leaves of a plant, stick it in our faces, and light it on fire. The nicotine in tobacco harnesses receptors that are for the endogenous ligand acetylcholine. So you probably heard more about that from Paul. I will not talk too much about it today, but the fact is that the kinetics of acetylcholine signaling and nicotine signaling differ quite greatly. Obviously, the clearance of acetylcholine is very rapid. There are mechanisms, particularly acetylcholinesterase, that breaks down acetylcholine very, very quickly after it's released. So its duration of action at the receptor is quite short. And there is some controversy on whether, for example, acetylcholine can actually desensitize nicotinic receptors. We know that nicotine itself can profoundly and long-term desensitize these nicotinic receptors, and that may be one of the big distinctions between endogenous acetylcholine and nicotine. And I'll talk more about that later. In terms of its properties at the receptor, the high affinity interactions between acetylcholine and the receptor and nicotine in the receptor actually are fairly similar, and it is much more the kinetics of release versus exogenous administration and breakdown versus clearance that probably are the big difference. So the receptors themselves have been the prototype for the biochemistry of ligand-gated ion channels now for probably 100 years. And one of the reasons is that it has been relatively easy to get large quantities of purified nicotinic acetylcholine receptors. Now, all of you know that all neuromuscular transmission, so all communication between the motor neurons and muscle, go through release of acetylcholine from the motor neuron and then nicotinic acetylcholine receptors at the neuromuscular junction. That receptor, which shares a lot of properties in common with the receptors in the brain, and I'll talk more about that, is very similar to the receptor that is found in an almost crystalline array in the torpedo electric fish electric organ. So I'm going to spend a second talking about this because this is one of the coolest biology things that is out there. So the torpedo electric fish, that's it right there. Actually, when I was a graduate student, Rick Huganier was working on the electric fish, and I'd sometimes come into the lab, and in the sink there'd be an electric fish. It would be kind of startling but very cool. They have two organs here that are specialized batteries, basically. So on one face of each cell, they're polarized epithelial cells. One face is almost exclusively an array of the sodium-potassium ATPase, so a sodium pump. And the other face is an almost crystalline array of the nicotinic receptors. And so can you see these little donuts here, these little flower-shaped things, all packed together? No, really? It may be hard to see in this light, but what you see is each of those dots is a pore of an of a nicotinic receptor. And if you could see the outlines, they're really these 5-petaled flower shapes that are packed one next to the other. So you could actually purify a lot of receptors simply by taking that battery out. So just to finish the story, when there is some stimulus, some touch to that electric fish, the acetylcholine release is massive and uniform across that array, and all of this array of receptors opens at the same time, and the charge that's been built up by the sodium-potassium ATPase flows across that entire battery immediately, generating up to 600 volts. Isn't that cool? So don't step on them. But as a biochemist, this is wonderful because you can actually purify a lot of protein, and the initial studies on the biophysics and on the sequence of these receptors was done by purifying them from the torpedo electric organ. So what do we know about them? Well, Nigel Unwin has been spending an entire career, probably 40 years now, getting higher and higher, incrementally higher resolution structures of the nicotinic receptor. More recently, actually, Pierre-Jean Coranger has been, and many others, have been crystallizing pieces of this, and particularly the acetylcholine binding pocket, which is mimicked by a protein called the acetylcholine binding protein that's an invertebrate protein. And so crystals of this portion at very high resolution have been made, but it's still been a challenge to get the entire transmembrane structure. But what you can see is the following. There is a large, this is the large extracellular space. The little lumps here are various subunits that are arranged around a pore. There's the pore. It's really pretty big. The vestibule is much bigger than the ions that it fluxes. But there's a constriction right here at the membrane. And this is where the bilayer would be. And at this constriction is where the selectivity filter for the ions that flow through this channel are, and where the gate for ion flux is. This structure was mapped onto the primary structure, and again, as a model of the ligand-gated ion channels, this is really the first, because it was also the first to be cloned. So at about the same time, Numa's group in Japan, Jean-Jeu's group in France took primary sequence from purified torpedo receptor and then cloned out the full primary sequence of the receptor, and the amino acid sequence was then mapped onto a hydrophobicity plot, and that was when they began to think about how the transmembrane structures were organized. So what you can see here is that there are 4 alpha helical segments of this receptor that cross the membrane, and these 4 helices form a sort of a coiled structure that is one stave on the barrel of the subunits that makes that pore. Now, here's what the my naive cartoon of these receptors looks like. You can see that there's the constriction, there's the gate, that's the pore. The ligand binding site for either acetylcholine or nicotine actually occurs at the interface between alpha and beta subunits. So here's the muscle, or the torpedo. Really, a lot of what we know I'm going to take from this. What we know is that you need an alpha and you need a neighboring subunit in order to get the ligand to bind. There are particular amino acids that are only found in alpha subunits, which actually defines an alpha. So if you have these 2 cysteines that can make that are fairly close in this primary structure, you call it an alpha. And for a long time, initially, people thought, oh, it's just the alpha. All you need to know about is the alpha. The others just sort of hold things in the right position. But if you look on the opposite side of that interface, there is another pocket. There are a few different loops that are conserved in the complementary subunit. And why am I making a big deal of this? Because when you have so many alphas and neighboring subunits, that means that the properties of that interface can vary greatly. So depending on which alpha you have and which neighboring subunit you have, you change the affinity of binding, the kinetics of the on and off rate, the likelihood of gating the channel, and therefore you have this really broad set of responses to either acetylcholine or nicotine that vary by subtype and by combination, and that can give you both short-term and long-term consequences of nicotine in the brain that can be traced back to really these fundamental differences at the amino acid structure at these interfaces. I'm going to keep talking about that. So there's the muscle type. There are two broad families of nicotinic receptors that are in the brain. There's this heteromeric type. It needs at least one alpha and at least one beta to make a functional receptor. And then there's homomeric type, which is really alpha 7, which can make a receptor with all alpha subunits. Now, these alphas are not the same as these alphas because this interface of the alpha has the domains that are conserved in the complementary, the complementary subunits. So this sort of hybrid subunit can act both as the left and the right side of that binding pocket. When these receptors were first described in the brain, they were described because the toxin, alpha bunker toxin, that had been used to study the muscle receptor was put on brain slices, and there were some spots. There was binding. And so this is probably in the 70s when that was first discovered. The initial reaction was, oh, that's an artifact, because we know that the nicotinic receptors are really important at the neuromuscular junction. Whatever that is, it's probably not important. It was not that long before people realized, wait a minute, there's nicotine in something that we actually take in through cigarette smoke, and there may be some reason to have receptors in the brain. But it's funny to think about the progress of science where the first reaction is, we study this in the muscle, it can't really be important in the brain. I think that's cool. So here's the evolutionary tree of the family. It's thought now that the alpha 7 subunit is probably the prototype, maybe the first type of subunit to evolve. You can imagine that making that first pentamer is probably very difficult. And so if you can do it with one type of subunit, it's likely that evolutionarily that's the most ancient, and then that the other family members really diverged from a primary subunit that could make homopentamers. This blue family here are the muscle receptors that I talked about. And then this family here are the subunits that need at least one alpha and at least one beta. And those are the heteromeric receptors. And then there's this family that are all related to alpha 7. Alpha 8 seems to be only in chick. Alpha 9 and alpha 10 actually form heteromers together. And they're highly expressed, for example, in the cochlea. Seems like there's a lot of nicotinic receptor in most of the sensory systems that have been studied. In the cochlea, they're essential for hearing and for development of the auditory system. They're in the olfactory system. In the retina, you can see the most dramatic array of expression of the neuronal nicotinic receptor subunits. It's quite startling how many receptors are expressed in the retina, and so on. So there's something about these receptors that has been fundamental probably to the development of these sensory systems. Okay, so then going back in history, again, the first time that a ligand-gated ion channel was actually reconstituted into a lipid bilayer, and we were able to see that you could actually evoke a current through these receptors. And this was done by Montal and colleagues. This was a lipid bilayer experiment where the purified torpedo nicotinic receptor was washed back into a lipid bilayer. And what you can see is there's sort of this leak current, right? Something goes through if you put a radiolabeled sodium on one side and you measure how much comes to the other side of the bilayer. And this is where they added acetylcholine. And what you can see is that there's immediate jump up in the flux of ions through that channel. And this was probably a remarkable moment. I can imagine sitting there and all of a sudden you see that your bilayer actually has this huge change in its properties. You make a hole by putting on the ligand. And that was really the beginning of understanding things like glutamate transmission, GABA transmission, all of the families of ligand-gated ion channels where the ion channel is an integral part of the protein really are follow-ons from these initial studies of ligand-evoked currents. Yeah?
No, the leak here, is that just an artifact of the system?
Yeah, it's just an artifact of having a bilayer, although maybe not entirely. So if you then go forward in a few years, which I will, and you start looking at single channel recordings, you'll see that there are flickers, right? So even without a ligand, there is a time that almost all, I think all, of these receptors will spend in an open state. It can be very brief. So it may be, in fact, that it's not purely an artifact of the system, and that baseline current is a property of the thermodynamic regulation of these channels. And we'll talk a little bit more about that now. So one of the things that has been really central to understanding these channels and the whole family of channels is the idea that they are not an induced fit molecule. So it's not that the acetylcholine or the nicotine comes in and forces the channel to open, right? We think about it as a ligand binding event that then opens the channel. So back in, I think the 1960s, Jean-Pierre Changeux and his colleagues Monod and Wyman. So Monod won the Nobel Prize for figuring out the lac operon. So all of us who use beta-galactosidase, we owe it to Monod and Jacob. But what Changeux did as a graduate student was he came up with the idea that, first of all, there is an allosteric mechanism for gating these channels. What does that mean? That simply means that the acetylcholine doesn't go into the pore and force it open. It binds somewhere else in the molecule, causes a conformational change, and the pore opens. I think we all take that for granted, right? I think we all consider, obviously, in a molecular structure, if you change some aspect of the structure here, you will change the overall conformation. But that was very revolutionary at the time. And again, just like in enzyme kinetics, where the idea that you don't actually induce the change by binding the ligand, but in fact you have a pocket that is in thermodynamic equilibrium between different conformations. And when you bind the ligand, what you do is you stabilize the conformation that, in this case, is the open state. So here's the idea. You have all of these states of the receptor. There's the resting state. It's not got a ligand. There is the open state that fluxes ions. And then there are other states where you can have ligand bound, but maybe it's not fluxing ions. And these would be this desensitized state and a more profoundly inactivated state. And in the normal course of a millisecond, this receptor may be flickering in and out of those states, probably spending much more time in one state or the other, right? So this is probably the most stable state without the ligand around. So when you get acetylcholine or nicotine in the binding pocket, what you do is that whenever it gets into this state, it stabilizes and it stays there much longer, right? So when you actually make this allosteric change, what the ligand is doing is keeping it more time in the state where ions flux. But because of the equilibrium between these states, this transition between the open state and the desensitized state is also happening all the time. And this state is even more stable than the open state. And I think that's central to understanding nicotine's effects in the brain. It's central to understanding the nicotinic receptor for sure. And that is that it really, once the ligand's on there, it likes to be here. It likes to be desensitized. And that desensitized state is something that is very well promoted by the slow, steady state release levels of nicotine, for example, in the nicotine patch. If you get low-level nicotine in the blood constantly all the time, what happens is that you never really pass through this open state for very long. This equilibrium is also occurring, and you're stabilizing this desensitized state. And it's likely that that contributes to the therapeutic action of nicotine patch, nicotine gum, other things that are absorbed fairly slowly through the mouth rather than quickly through the lungs where it gets very rapidly to the brain. So how do you actually study this state? Actually, I'll not get to that yet. But the idea that these states actually have different affinity for the ligand is important as well. If any of you work on G protein coupled receptors, you know that the desensitized state actually has lower affinity for the ligand, so it gets kicked off. And so there's a cycle. Okay, you have the resting state has pretty high affinity for the acetylcholine. Let's say it's a G protein coupled muscarinic receptor. It binds, it is activated, and then once it's activated, it actually has a lower affinity for the ligand, so the ligand can come off and you can start the cycle again. Nicotinic acetylcholine receptors are the opposite of that. That is that these desensitized states actually have much higher affinity for the ligand and therefore hold on to it very tightly. And so the equilibrium between the on and off of the nicotine in these desensitized state is much slower than in the resting state. And again, what that just says is that once you have nicotine in there, the transition back to an open, active channel is really not that favored. Okay.
Marina,
Yeah.
I've always been interested.
Yeah.
Do you think that's an artifact of the fact that it's got to do some work to do conformational change? So the actual initial affinity may be very, very high, but then as soon as it's done as soon as it's doing conformational change, then that energy of binding is transferred. So I think there may be a very, very high affinity right at the beginning, and then what people are measuring is that change of energy of binding to a conformational shift. I think it may be almost impossible to...
Yeah, I think it's almost impossible to measure that, although there have been some attempts to do that by Changeux and Edelstein in making their models. And I think that capturing those moments, those transitional states, is impossible. Maybe you could do it with a ligand, which is what they've tried to do, that traps it halfway. Yeah.
So from like a functional standpoint, if
Yeah.
you think of a neuromuscular junction, what benefit, when you go to the desensitized state, and then it's even harder to recover, so that means recovery is really extended. If you're going into a high affinity state, you need the acetylcholine concentration to drop
Right.
tremendously. So from a functional standpoint, why do you think it's evolved to function this way?
That's a great question, and I've wrestled with that a lot because it is not entirely clear, for example, at the neuromuscular junction, that the acetylcholine ever reaches some sufficient level to desensitize the neuromuscular receptor. That may be because it is cleaved so rapidly that very little actually makes it across to the receptor from the release site. In the brain, there are probably two modes of acetylcholine action at these receptors, and this is really, optogenetics has begun to tease this out. It was not appreciated that the brain was very different from the neuromuscular junction. It was thought that you have synaptic release, you have a lot of acetylcholinesterase, so very little escapes the synapse, and you have this very rapid event that perhaps did not desensitize. What is clear is that you have that in some cases. It was very hard to find the direct synaptic effects, and in fact, they're fairly rare. You can definitely find them now, both in hippocampus and cortex. More often, it seems that in the brain there is volume transmission through acetylcholine onto nicotinic receptors. And what does that mean? That means you have release that escapes the synapse. We don't really know for acetylcholine how far that can go because the levels of acetylcholinesterase are still very high. But that sort of transmission seems to define, let's say, a state of a system rather than a transient activation of that system. So the way that I think about it is that let's say that something in your environment alerts you that something is important. You definitely get activation of both the basal forebrain complex that releases acetylcholine as well as the brainstem nuclei, the pedunculopontine and the lateral-dorsal tegmental nuclei. You get a lot of acetylcholine released. You, in fact, switch their firing rate to increase release. And then you have your nicotinic receptors that I'll, I think, show you later are not just on cell bodies or postsynaptic. They're on the axons. They're on the terminals. And that entire neuron may, in fact, now have a higher basal rate of nicotinic transmission. Is it possible that over time, if you got high enough levels of acetylcholine, that you would want to be able to have feedback to shut it down in order to prevent, for example, the kind of excitotoxicity you get with glutamate? I think that's likely. The levels are so low that I think you rarely reach that with acetylcholine. But if you had an injury, if you had some event that perhaps raised stress for long enough, then you would actually, we know that, for example, if you put a constitutively active nicotinic receptor genetically into neurons, you get progressive degeneration. It's not rapid. It's actually slow over the course of development and adulthood. But clearly, that feedback would decrease the likelihood that you get excitotoxicity.
Yeah, the way I was thinking about it, the neuromuscular junction, it didn't quite make sense to me because
Yeah.
I thought, well, okay, you're going to go from something like a muscle spasm
Right.
to paralysis.
Right. Which would not be so good. Yeah,
Right, so
exactly.
but if they never actually get desensitized.
So at the neuromuscular junction, there is controversy, but the consensus, I believe, is that really don't get desensitization from acetylcholine. But with nicotine, it's the opposite. And that is where we don't have the rapid clearance, where we have liver, basically it gets, nicotine gets degraded by going through the CYP450 enzymes in the liver. And it gets metabolized to cotinine and then cleared through the urine. If you have nicotine in your brain for a really long period of time at levels that are more than sufficient to drive it into the desensitized state, you actually have an extremely long recovery in between bouts of open states. Okay. So actually that transitions very well to one of the key differences that I think is really just in the process of emerging. It's been talked about for a long time, and that is that alpha-7 homomeric receptors actually behave differently than these heteromeric receptors. In oocytes, when you reconstitute these receptors and you look at their responses to acetylcholine, their responses to nicotine, alpha-7 was always thought to be extremely low affinity for acetylcholine and nicotine. You needed a lot to get the maximal current, but that it desensitized really rapidly. So once you put a lot of nicotine on, it just stayed in that desensitized state for a very long time. And in fact, the prototype for all of those kinetics is alpha-7 because it's easiest to study. So it may be the poster child for something that once it's fully grabbed onto nicotine and in the desensitized state, it really has a high affinity for nicotine in that state. But then many people, Lorna Roll's group, Dan McGee's group, John Danny's group, were doing in vivo slice recordings, and they were looking at the effects of very low levels of nicotine on persistent presynaptic potentiation of other neurotransmitter release. So for example, on glutamatergic terminals, there are alpha-7 receptors, and what these folks found, particularly Dan McGee's lab, is that very, very low levels of nicotine could still continue to cause nicotine-induced glutamate release in that slice. So they also knew that the pharmacology of that response was this homomeric alpha-7 receptor because they could block it with antagonists of alpha-7. So how do you reconcile the fact that in oocytes, these receptors are low affinity and very rapidly desensitizing, and in a slice, you could use very low levels of nicotine and that they were not desensitizing? And really, I think it comes down to the fact that we were looking for the wrong thing. In oocytes, physiologists were looking for the peak current. When do you get the maximal response? In vivo, that probably almost never happens, if ever. And that's because you need very high levels in order to get all 5 interfaces of the alpha-7 receptor occupied. So the low affinity responses that they looked at in oocytes were when you get every single one of those interfaces occupied by nicotine or acetylcholine. Then you get a really big open burst of that receptor, all the receptors in the oocyte, and then they shut off because they're grabbing onto that ligand. In vivo, when you use very low levels of nicotine, so the kinds of levels of nicotine you might get from a cigarette smoke, a cigarette puff, you would probably not occupy all of those. In fact, you might occupy 1 interface, maybe 2 in the way that you would in a heteromeric receptor. But at that point, there would still be other interfaces available, even if those 2 interfaces were occupied. So you might be able to come in with another puff of low-level nicotine and still open the receptor. And the likelihood of driving this into the desensitized state or stabilizing the desensitized state might be much lower. So I think it's a cautionary tale for what we actually want to look for in vivo compared to in vitro. We have to remember that the basic fundamental properties of either a molecule or a cell in vitro can be something that we can establish, but those parameters may not be the same that operate in a slice or in a whole brain.
So this puts a big problem with imaging studies. How do you interpret them? Because your affinities
Yes.
are changing in the desensitized state.
So I have an enormous problem with PET and SPECT studies of receptors because there is a competition between the endogenous ligand and the very, very low level of the tracer. So that's one problem. It's a little different from the one you were just pointing out. But when we do receptor binding to a ligand in a slice, what we do is we wash out the endogenous ligand before we put on the radioactive ligand to say, okay, how many receptors are there? What are their properties? You can't do that in a brain. So any imaging study with a radio-labeled ligand, you have to ask yourself, is the difference that I'm seeing a difference in the number of molecules, or is there a competition between whatever's the normal ligand for that site and the tracers? That's one problem. And it's a big one, frankly. I think that if you make the conclusion that drug abusers have more or fewer D2 receptors, you have to know whether there's also more or less occupancy of those receptors by their endogenous dopamine ligand. That's important. The way to do that is to confirm it with postmortem studies, which has been done. But the second issue is, if you give the ligand and there's enough of it to change the state of the receptor and to shift it from a lower affinity to a higher affinity state, you can actually be measuring only the desensitized receptors, or you could be actually increasing or decreasing the number of those desensitized receptors. That is also a problem with other receptors, but probably most for this receptor, where the affinity increases with desensitization, paradoxically. So I just want to point out here before we go on, so there's the alpha and the beta subunit, there's the interface, and if we take the analogy that you could make the alpha-7 act like a heteromeric receptor, then maybe this double liganded state actually acts much more like this heteromeric receptor. And this is the weird one that we measure in oocyte studies. Okay, so now we're going to dig down even a little deeper to these loops that I was talking about. And what you see here is that the alpha subunit here has a few different loops that would bind the neurotransmitter, in this case acetylcholine. And the neighboring structural subunit, it's beta in the heteromeric receptors. It's another alpha-7 in the alpha-7 receptors. It has another stabilizing loop, loop D, that's really essential for whether you have a high affinity interaction, a lower affinity interaction, and then the off rate. And what you can see is that these loops are conserved not only in the nicotinic receptors, but also in the ionotropic GABA receptors, so the GABA A receptors, in the glycine receptor, and in the one serotonin receptor that's also got an ion channel. So this is probably a family that evolved from a similar prototype. So what happens in the pore? One of the nice things about the recent imaging studies with the acetylcholine binding protein, which is really that outside ligand binding domain, is that we can actually see what changes inside the receptor as these processes occur. So the model is that all of these subunits are sitting there with a kink in their the long axis of the molecule, and that kink is that narrowing that that is the filter in the pore. And that what happens when a ligand binds is that they basically all switch together and tilt so that that portion that causes the constriction is tilted outward, and the pore opens, allowing the ions to pass. So that is something that you can determine with site-directed mutagenesis, with binding of various ligands, and then isolating the protein and looking at the particular amino acids that interact with, for example, pore blockers, channel blockers versus blockers of the acetylcholine binding site. And as a result, we have this model here of these rings of amino acids that form various filtering and constrictions of this pore. So if you mutate this ring here in light blue, you can shift the selectivity of the alpha-7 type receptor from a single positive ions, so sodium, which only has a single positive charge, and you can actually switch between calcium and sodium gating. So that's pretty important. You can imagine that if you had an NMDA receptor that had a similar structure, which they don't, by the way, the glutamate receptors do have a slightly different structure. You could shift between a receptor that fluxed calcium and one that simply depolarized the neuron. And that seems to be the case between, for example, the heteromeric receptors that are good at depolarization and the alpha-7 receptors that get more calcium into the cell. Then you have this loop here, and you can see that sizes of ions matter. So rubidium, which is used as a sodium analog, can actually pass through most nicotinic receptors, all that I know of, but this loop can be mutated so that you only have rubidium or sodium flux through the channel. Here's another calcium versus sodium gate, and then this is really cool here. This proline and then these glutamates right here adjacent to the proline. If you look at the GABA receptors, which flux chloride and inhibit, that hyperpolarize a cell, versus the nicotinic receptors that depolarize and excite the cell, the big difference is in these two loops here. So you can just mutate one proline and one glutamate, and you can shift a depolarizing channel into a hyperpolarizing channel. And that's here right at the bottom of the pore. So one of the things that was really essential in getting at this model of allosteric modulation, of these loops being important for gating various states of the receptor, was this experiment here. And this was done by Jean-Luc Galzi in Jean-Pierre Change's lab in the late 80s, I think, maybe early 90s. And what they did was to systematically mutate the residues in those loops. So they'd identified them biochemically. Now they went in, they tried to get a nice mechanistic basis for their theories about these loops. So they went in and they changed these various residues, and then they looked at the properties of these receptors in either an oocyte or a bilayer. And one mutation really stood out, and it has been essential for now going into animals, and that's why I'm focusing on it here, because it's really given us a good idea of what these channels normally do in the systems that are important for addiction. So this leucine here was thought to be very important for the desensitization of the channel, so they mutated it to a threonine. And what they saw was the following. So here is a measurement of the current when you put acetylcholine or nicotine onto this channel. Normally, you get a very rapid opening. This is an alpha 7 type channel. At high concentrations, so we're talking about 100 micromolar acetylcholine. That's a lot of acetylcholine. You don't get that very often. That's probably why we don't drive it into the desensitized state. And then it desensitizes, so even though the ligand is still there, it closes. You have some little steady state current, and then you take away the ligand and it goes away. That's normal. What happens when you make this one mutation? Well, you get this very, very slow current that stays as long as you have the ligand around, and you need very, very low levels of acetylcholine to get that current. So what do we know about the receptor from what I said? The desensitized state is very high affinity. You need little acetylcholine to get it. It should be a stable, long-lasting state. So what they hypothesized was that, in fact, they had turned that ring that usually is closed in the desensitized state into a ring that is open during the desensitized state. And so now, with very, very low-level nicotine, acetylcholine that would normally desensitize the channel and cause it not to flux ions, they got a whole massive current, just as much as if they put 100 micromolar, and it stayed there as long as the ligand was there. And so what does that mean for the receptor? It means that in vivo, any low level of choline, which is also an agonist for the alpha 7 receptor, or acetylcholine would actually turn on this receptor and keep it on for a long time. And it turns out that if you put this mutation into, you knock it into the alpha 7 subunit of a normal mouse, you now get massive hippocampal degeneration, progressive cortical degeneration, and the animals die. And it's thought that that occurs because choline is actually now an agonist. There's a lot more choline out there than there is acetylcholine. What cholinesterase does is it breaks acetylcholine down to choline, and choline is essential for making lipid bilayers, so there's a lot of it around. It actually signals, and now that receptor is, because it's fluxing in this desensitized state, is causing excitotoxicity. But it also tells us a lot about the structure of this receptor, and it says that that loop probably is important for this allosteric transition between an open state and a desensitized state. So now I'm going to jump to the in vivo imaging that we just talked about as being problematic. And this is an image through the human brain going this way, where the thalamus is loaded with nicotinic receptors. And this is a SPECT study with a nicotinic ligand that binds almost exclusively to the, as far as we know exclusively, because we can't see any binding at all in a knockout, to the highest affinity heteromeric receptors. So these receptors are found throughout the brain. In fact, it's very hard to find a negative area in the brain to use as a negative control. So its specificity of this ligand is based on the differences in region rather than the fact that you can say, here, this region's completely blank. If you look at the pattern of binding, so here's the human brain this way, and I apologize, the mouse brain is this way, you can see that the hot spots are the same. There's the thalamus in the mouse. There's the cortex in the mouse. And you can see that you see general binding of these high affinity receptors really throughout the brain. There are a few neurons that don't respond to nicotinic agonists. One happens to be the medium spiny neurons in the striatum and in the nucleus accumbens. That's kind of interesting. All the inputs to those neurons have nicotinic receptors, but the outputs, not so much. And there are probably others that just haven't been discovered or reported, because when you get a negative result, it's harder to publish a paper on it. But what that says is that the distribution of nicotinic receptors across species is actually fairly highly conserved. And one of the key things that has been seen in human brain from smokers or in rodent brains that have been treated chronically with nicotine is that all of this allosteric stuff matters because it changes the number of nicotinic receptors if you've had chronic nicotine exposure. So here is postmortem cortical tissue from a non-smoker and from a smoker. There's the background. You can compete it away with nicotine, and what you see is there's about a doubling of the number of these high-affinity nicotine binding sites in the brain of a smoker. And the same is true if you treat a rat twice a day with nicotine for 10 days, or if you give a mouse nicotine in their drinking water for 30 days. Probably all of this has to do with how much gets into the brain because of the metabolism of the various species. And what that means for yeah.
And can you is there a difference, like a different part of the brain about that?
Yeah, so there are some differences. For example, in the thalamus, where you have such high levels to begin with, you don't get very much upregulation in a smoker or in a rodent. Whether that's because you're already, well, I'll tell you a little bit more about why we think that this upregulation occurs, but it is post-translational. That is, it doesn't occur at the level of the RNA. It's not that there's more receptors being synthesized from the at the RNA level. In fact, what probably is happening is that we know nicotine gets into the cell itself. It crosses across, it goes across the membrane when it is in basically in a basic gives up the protonated state, deprotonated. When it is neutral in charge, it crosses across, and then inside the cell, it actually becomes charged and it gets stuck. And when it is inside the cell, it can actually bind at the interface between the subunits in a heteromer. And when it binds, we know that making a 5-protein complex is really hard. And all of that occurs in the ER as these subunits are being synthesized. There are chaperones that aid in this process. There's RIC3, there's probably 1433, which are another class of chaperones. They probably hold those subunits in a form where they can actually get together and bend into this barrel. But when nicotine gets into the cell, you can imagine that when it binds at the subunit interface, it probably also acts as an intracellular chaperone. And then it also allows that receptor to be trafficked out of the ER more rapidly to the Golgi. What may be the case in the thalamus is that that process is optimized already with a set of chaperones that are not expressed in other brain areas. And that's an area of pretty intense study. So maybe you're at your max, and so getting nicotine in there doesn't actually speed it up at all. But we do know that the processes that occur inside the cell with nicotine are occurring actually at the level of the already translated receptor.
So you're sure that's not a change in affinity state?
It is not a change in affinity
It's gonna
state.
be
Yeah, because you can do it in postmortem tissue. You couldn't know that if it was in a brain. What you can do is you can saturate it and you can push everything to the most desensitized state, and then you can actually get a question. You can measure number, and you can also at this point now do it with mass spec, and we've done that with a proteomic study from both mouse brain and human smokers, and you actually get twice as much protein coming out in the peaks of the peptides that predict each subunit.
So it's every subunit? Or is it just
No, so at least so the ones that are most susceptible to upregulation are those containing the alpha 4 and the beta 2 subunit. If you look in vitro in cell preparations, you can also do it with the other heteromeric receptors, the beta 4 and the alpha 3. Alpha 7, not so much. There are certainly reports of upregulation. There are also some reports of downregulation. It's not the same process. Maybe because it's, and this is a just so story, this is hand waving, but maybe if you need an alpha to find a beta, it's a more difficult process than it is for an alpha to find an alpha if they're made at the same time from the same RNA. I don't know if that's the case. So one of the things that's been really a question is for smokers who quit smoking and have twice the number of receptors in their brain, does that contribute to their relapse? Does it contribute to craving? There are a couple of imaging studies now that have correlated the binding of this ligand after washout of the endogenous nicotine, because it competes with the ligand, with scores of craving. And again, it's mixed. There does seem to be some, there are some studies that report a correlation between the number of receptors in the quit smoker, the former smoker, and their self-reported craving on craving scores. So there may be something there. You can imagine that if you had low levels of nicotine from smoking in your brain at all times and you were desensitizing your receptors chronically, and then that got washed out during abstinence, having twice as many receptors might in fact change, and certainly would, change the dynamics of acetylcholine signaling in the brain. So there's reasons physiologically for this potentially to happen. Whether this contributes to relapse, we don't actually know for sure. The thing that we do know is that it's a very slow recovery process. So the half-life of these nicotinic receptors is at least 2 weeks in all of the in vitro systems where it's been studied. So once you have double the number of nicotinic receptors, they actually stay up at least for 6 weeks. I think this is at 30 days. I've forgotten, but this is a study from Julie Staley and her colleagues, and they looked over time at abstinent smokers and saw that even weeks after abstinence, they still had more receptors than a non-smoker. So what do we know about these receptors in particular brain areas and particular neurons? Well, one of the things that really stands out when you do a ligand binding study that looks at the high affinity binding sites, and again, I mentioned those are alpha 4 beta 2, the subtype I'll talk about a little bit more in a few minutes, is that the VTA and substantia nigra are loaded with nicotinic receptors and with an abundance of different nicotinic receptor subtypes. You can't see alpha 7 here, mainly because I think this exposure is a little bit low, but if you do single cell RT-PCR or if you stimulate these VTA dopamine neurons, there are alpha 7 subunits on those neurons as well. This ligand is actually, this is in situ, so each of these slices is with using a different in situ probe that's very selective for a particular subunit. What you can see is that there are subunits that really particularly target these catecholaminergic neurons. If you looked at these subunits, alpha 6 and beta 3, you would also see that the noradrenergic nuclei are loaded with alpha 6 and beta 3. They're also in the retina, because I told you everything's in the retina. But what that has raised as an exciting possibility is if you could actually target the alpha 6 or the beta 3 containing nicotinic receptors, would you have a more selective treatment that really targeted their effects on the dopaminergic neurons rather than all of the other neurons that also have high affinity nicotinic receptors throughout the brain. Alpha 6 and beta 3 act as secondary accessory subunits. They can't form receptors on their own, but they combine with alpha 4 beta 2 or alpha 3 beta 4 to make a receptor. these subunits, beta 2 and alpha 4, are extremely widespread, and they really are the core of the binding site that I showed you for the high affinity nicotine binding. You can see that they're there at fairly high levels in the VTA and substantia nigra, but they're also everywhere else, right? So lots in the cortex, as I mentioned, and I'm not going to show you here, but there's a lot of these receptors in the thalamus that constitutes that very high level concentration of nicotinic receptors. And then there are some subunits like the beta 4 and the alpha 3 subunit that are actually fairly, they are expressed, it turns out, at very low levels in other brain areas, but they have hot spots of expression in, for example, the medial habenula, which I hope Paul Kenny talked about, I will talk about for a minute here, just to reiterate what you heard from him. And what's really interesting about those subunits is they are responsible for all the cholinergic transmission in the autonomic ganglia. They're essential for the sympathetic response or the parasympathetic response to threat. If you don't have those beta 4 alpha 3 receptors, you don't have a functioning autonomic nervous system. So we also have pockets of expression of very high levels of those subunits in other parts of the central nervous system, particularly medial habenula, interpeduncular nucleus. And there's low levels of expression in hypothalamus, for example. And what I like to imagine is that there's some coordination between the peripheral autonomic response to threat and the central hotspots for these autonomic receptors. And I'm wondering if there's some coordinated response to, for example, aversive stimuli or other environmental threats that explains what these beta 4 and alpha 3 receptor responses are doing in the brain. So they act as, in some ways, an accessory system that may limit the activity of the dopaminergic neurons, certainly act in parallel and perhaps in an opposing manner. And as Paul said, that can actually be very influential in how much nicotine you would self-administer. So the brakes are just as important as the accelerator, maybe more so. Okay, so what happens on the dopamine neurons themselves? We know that there are nicotinic receptors on almost every aspect of this circuit. On the cell bodies, there are receptors containing the alpha 6 subunit, containing the alpha 4 subunit, beta 2 subunit. And if you put nicotine onto a slice preparation and you look at the firing rate of these dopamine neurons, there is a postsynaptic nicotinic receptor. Postsynaptic just meaning that it is not on the terminal. It doesn't necessarily have to be opposed to a site of acetylcholine release. But those receptors increase the firing rate of these neurons. There are also nicotinic receptors on the terminals of these dopamine neurons, so that if you make a synaptosome preparation, if you just have terminals and you put nicotine onto those terminals, you get nicotine-induced dopamine release from those terminals. And that seems to be very important in the nucleus accumbens and striatum. There have been some very nice studies originally by Margaret Rice and Stephanie Cragg. Stephanie's continued to study this really extensively, also by Dave Selzer's group, that showed that acetylcholine inputs to the, from those actually tonically active large cholinergic interneurons in the striatum and accumbens provides a fairly continuous level of acetylcholine to these terminal nicotinic receptors. So there's a low-level basal response, dopamine response to the tonic firing of those acetylcholine interneurons, and that when you block that cholinergic signaling by putting a nicotinic blocker, or presumably by pausing the firing of those cholinergic interneurons, that you reduce the basal levels of dopamine release. You can do that with voltammetry, you can do it with amperometry. And what's important about that is that when we perceive a reward or a novel stimulus, one of the things that's going to be important is not only the size of the signal, but the level of the noise. So if those cholinergic interneurons pause, the noise goes down, and that signal rises up out of the noise, and the difference between signal and noise is much greater. So at baseline, the chronic stimulation of those receptors, they don't seem to desensitize, does seem to provide a low-level stimulus in vivo of those terminals, those dopamine terminals. So I think that's pretty cool. So what is, of course, true about this system, like any brain system, is that it is not that simple. There are nicotinic receptors, as I think I mentioned, on the terminals of the glutamatergic inputs to those dopamine neurons. Those seem to be largely alpha 7. Certainly that's what I mentioned earlier from slice preparations, where you can put low levels of acetylcholine or low levels of nicotine and get an an alpha 7 mediated glutamate input to those dopamine cell bodies. That's not only important for boosting their firing rate, it's important for inducing long-term potentiation of those connections. So the glutamate release actually potentiates the synapse between these glutamate neurons and those dopamine cell bodies. And so overall, this drive is probably extremely important for longer-term consequences of nicotine, and that's been shown at least in a slice over hours from Dan McGee's lab, where he's shown that you can get LTP at this synapse that allows the glutamate signaling to continue to drive dopamine neuron firing even after the nicotinic receptors themselves should probably be desensitized. As I mentioned, the acetylcholine, the tonically active cholinergic neurons in the accumbens and striatum are stimulating those terminal receptors. But then you've got the added complication of GABAergic neurons, which I think may be really at the center of the ability of nicotinic receptors to modulate the circuit and to change its activity in the as a consequence of smoking, and possibly to drive long-term addiction as well. So it contributes for sure. So these GABA neurons have the same high-affinity nicotinic receptors, not the same. They have high-affinity nicotinic receptors on their terminals, and they're impinging on these dopamine neurons with high-affinity nicotinic receptors on their cell bodies. These are a little different because they contain the alpha-6 and beta-3 subunits. Remember I told you they are really very selective for the dopamine neurons. I may introduce a complicating comment in a second. And then on the terminals, you have these high-affinity receptors as well. When you, in a slice, bathe the slice with low levels of nicotine for a long period of time, these get desensitized, but these continue to drive. So that's where this balance of how much nicotine at that interface actually can cause the allosteric transition and open the channel rather than drive the receptor into desensitized state actually matters. Now, here's the complicating thing. There's one paper out there that says you can dissociate VTA neurons and put nicotine onto those dissociated VTA neurons, and through an alpha-6 mechanism get GABAergic release, GABA release. Now, I was very puzzled by this, and I thought it can't be right. We know from single-cell RT-PCR, we know from in situ hybridization, there's an almost strict limit of alpha-6 to these dopamine neurons. And then a really beautiful optogenetic paper came out from Bernardo Sabatini's lab about a year ago that showed that dopamine neurons can actually take up GABA from the environment through the monoamine transporters. And it can actually release it even though those dopamine neurons don't make it themselves. His theory is that this is a way for those neurons to sample their environment. So if there's been a lot of GABA released, you're now going to have a very different response when you stimulate those dopamine neurons than if you would if there had not been GABA released previously. I think that alpha-6 paper was seeing this in cultured neurons, where there is actually a fair, there are both GABAergic and dopamine neurons in those cultures. There's probably a lot more neurotransmitter in the extracellular milieu in those neurons. I'm guessing that they saw the fact that dopamine neurons are not just dopamine neurons. I think you've heard about the fact yesterday that they can also release glutamate, and that there are subsets of neurons that are glutamatergic in VTA, subsets that are dopaminergic, subsets that are both. The same is true for GABA. But this other mechanism actually isn't because those neurons are GABAergic. There are also GABAergic neurons there, but that the dopamine neurons can also take up and release GABA from the extracellular milieu. Just a complication. I apologize. So,
when you say release, like, is it?
It's vesicular.
Co-release, like what
is it co-release? That is not entirely clear yet, I think. When they do the optogenetic stimulation, they see a massive, faster GABA release than they do, yes. And so you can see these inhibitory postsynaptic responses that are very fast. Whether that's because it's a different set of vesicles or whether it's because they're only measuring the sort of swamping consequence of the fast-acting neurotransmitter, I don't know.
If it's the same transporter, there's reason to believe it's the same vessel.
Right. Although, if there's a lot more GABA, you could imagine that it might in fact outcompete dopamine in some synapses. Hard to know.
Yeah, it's hard to say.
Yeah.
If they're both VMAT dependent,
Yes.
there's no reason to think they couldn't be co-packaged. There has to be something
Yeah.
else to distinguish.
That makes sense. There you go.
I would probably.
But what I think is exciting is that it really adds another level of computation to that circuit. So I really already talked about that. The fact that chronic nicotine would then desensitize this GABAergic release onto these cell bodies. It seems to leave the alpha-7 response pretty much intact, probably because we're talking about a fairly low level of nicotine, particularly from in the slice experiment where it's measured, but also in the brains of smokers, so that you get a mix of excitatory and inhibitory drive onto these neurons at baseline that shifts to a more unmixed excitatory drive. And desensitized.
How do you think the interaction with the THC system and the opiate system is?
Oh, that's such a good question. So, you know, the idea that disinhibition of the GABAergic neurons is central to, let's say, cannabinoid or opiate transmission in the VTA is one that's extremely attractive, but I think that the desensitized nicotinic receptors, if there's some tonic stimulation by acetylcholine of those nicotinic receptors, could do something quite similar, right? So if you have a baseline tone through acetylcholine that's removed by desensitizing those receptors, maybe you actually can also decrease GABA in the same way with chronic nicotine that you do to some extent with cannabinoids and opiates. So that if you and if you ask the question, is there a molecular interaction between nicotine addiction and the action of those opiate and cannabinoid pathways, which there clearly is. I mean, there's obvious differences in knockout mice that lack opiate receptors. Or
Maybe you guys don't know this story yet, so maybe we should
I will tell the story. So what I'm about to tell you is that you need nicotinic receptors for nicotine actions really right at the beginning. That's not so surprising. If the nicotinic receptor is the primary molecular target for nicotine, if you can't actually transduce that signal, it's not so surprising that you don't get some of the hallmark behaviors of reinforcement and so on. But knockout mice that lack, for example, the mu-opioid receptor, the CB1 receptor, also show greatly decreased nicotine reward. So you can measure that in place preference experiments. You can look at even locomotion. And you can see that there is an interaction between these systems. All of these drugs are abused. Sometimes, maybe often, they're co-abused. So what I think is our conversation was about is where in the circuit is this happening? And the answer is we really don't know. But one possibility could be right in these GABAergic neurons in the VTA where, in a sense, desensitizing nicotinic receptors would have similar effects to stimulating cannabinoid receptors or stimulating opioid receptors, both of which are G protein coupled receptors that are coupled to GI and would decrease firing of these GABA neurons. Desensitization of the nicotinic receptors could only work there if there was some normal tone that was acetylcholine, for example, that was stimulating those the nicotinic receptors on GABA neurons. I think that's a likely scenario, but I don't think that we have enough data to say. We're working on that. Not particularly the cannabinoid and the opioid receptor interaction, but I think we could long term. But right now, asking what is it that nicotinic receptors are normally doing in response to acetylcholine in those GABA neurons, and what happens with chronic nicotine? What's the difference between the receptors on the different cell types in the VTA? We know VTA is critical. You can locally infuse nicotine into the VTA, and Roy Wise and colleagues showed that you get place preference just fine. Ikemoto and colleagues showed that you'll self-administer nicotine right into the VTA. So VTA is enough. You ablate VTA, Paul Clark and Bill Corrigal showed you get no nicotine reward, as you would expect from other drugs of abuse. VTA is the hot spot. You absolutely need VTA to start this. But which cell types in the VTA are absolutely essential? We know dopamine neurons are essential, but there's emerging evidence that the other cell types also are essential. That is, there's synergy between the cell types and VTA. Are there other systems that are also absolutely critical for human smoking? No question. The medial habenula interpeduncular pathway, the cortical pathways that also impinge to result in inhibitory control or the lack of inhibitory control, also important from the standpoint of nicotinic receptors. But the VTA is really, really important, as you probably all know. All right. So now I'm going to get into these experiments, the knockout mouse studies that really implicate, I'm going to finish pretty soon, I'm sorry to go over, that implicate specific receptor types and specific neuronal types. So we know that the alpha-4 beta-2 receptors are widely expressed. They're expressed in VTA. Alpha-3 beta-4, that's one of the hotspots. That's the interpeduncular nucleus. And what you can see is that you can knock out one of these subunits, leave the others completely intact. So if you have a wild-type brain and you look for ligands that either selectively find the high-affinity receptors or find all of the receptor subtypes, you can get rid of the high-affinity receptors, for example, without getting rid of the other subclasses. You can actually start to dissect out the roles of the different receptors. We didn't know that to start with. We thought that it was possible that getting rid of one subunit could alter all the other nicotinic receptors. You could get upregulation, you could get shifts in their expression. That does not seem to be what happens. And the same is true for alpha-7. If you get rid of the high-affinity receptors, alpha-7 really still stays the same. You can see a lot of alpha-7 in regions of the brain involved in cognitive processing, a lot in the hippocampus, a lot in the cortex. I think that's important. So if you knock out only one subunit, the beta-2 subunit, you actually completely abolish that postsynaptic response on dopamine cell bodies. Even though there are multiple receptor subtypes, you need that core beta-2 subunit to get those neurons to fire in response to nicotine. Even low levels of nicotine, they're a little higher than would be in a cigarette, still see an inward current in a wild-type slice, not in the knockout. You increase the firing rate, and that goes away in the knockouts. And that translates to the whole organism. You can give nicotine peripherally, and despite the fact that nicotinic receptors are found throughout the brain, these intrinsic responses of the VTA dopamine neurons are not driven, for example, by that nicotine-induced glutamate release. Without the postsynaptic receptor, you don't get those neurons to fire, and you can tell that because if you do a microdialysis study, if you don't have those high-affinity receptors, having the alpha-7 is not enough to rescue. And then you can look at behaviors that I'm sure you've already talked about. So in rodents, we have some models that are try to dissect out different aspects of the rewarding and reinforcing properties of various drugs of abuse. But you can also use them as assays of function of various brain areas. So locomotion, for example, we know is a consequence of stimulating the dopamine levels, release, or activity of the dopamine neurons. So you can use it as an assay of the ability of a ligand to stimulate the dopamine neurons. And here what you can see is that normal mice will get actually a locomotor response, an increase in locomotion in response to nicotine administration, and that you can get rid of that by abolishing, by knocking out a receptor subtype that's expressed on those dopamine neurons. Similarly, place preference, at least to a first approximation, may model the ability of a drug of abuse to cause an association with a particular set of stimuli and some kind of rewarding or reinforcing signal. And what you can see is that nicotine can induce a place preference in mice. I can tell you that it is not easy, that it is definitely very, very strain dependent. It is stress dependent. It is something that is very, very tightly dose dependent, so you have to find the exact dose. But when you get that dose, you can get a preference in mice, and it may be that that exquisite dose sensitivity is why smoking is such a good drug delivery device. It is a way for you to very quickly titrate the amount of nicotine that gets to your brain. First of all, because smoking gets the nicotine to your brain very rapidly by getting the nicotine down into your lungs and directly up into the brain without passing through the liver. But also because you can stop at any time. As soon as it feels aversive, you just don't take another puff. And I think that exquisite dose sensitivity really complicates studying nicotine reward and reinforcement in rodents, because you want to have a system like self-administration where it is also directly under the control of the animal how much they get. And you can get mice to self-administer nicotine. In this case, you can train them on one drug and then switch them to nicotine. They'll continue to press for nicotine, and that response is extinguished when you switch to saline, or it's extinguished if you switch animals without high affinity nicotinic receptors from cocaine to saline. So all of the hallmarks of the most basic models of drug response are gone if you get rid of the nicotinic receptors that have the highest affinity for nicotine. Makes sense? You need high affinity responses in order to get to transduce the signal from a cigarette, which doesn't get that much nicotine to the brain. And it makes sense that if you don't have the receptor, you don't actually get the behavioral responses that initiate addiction. But I want to stress that this would be the initiation, right? These would be the first steps. Doesn't mean that these receptors are either solely responsible for addiction to nicotine, or that the long-term responses only depend on this receptor subclass.
They are well aware of the dark side.
Yes, you know the dark side. And so going back to that mutation where you can make the receptor basically much, much higher affinity by making the desensitized state a fluxing state, you can put this L247T mutation into any of the nicotinic receptor subunits, and you can get a receptor that is more or less activatable. And Henry Lester's lab were pioneers in this area, and they put these mutations both into the alpha 4 subunit, so that's the high affinity receptor that would be lost when you knock out beta 2. They also put it into the alpha 6 subunit that should really be concentrating this receptor subtype, the activatable receptor subtype, to dopamine neurons. And so what they saw was that if you knock in that desensitizing mutation, what you see is that you get currents at levels of nicotine where you really don't see anything in the wild type. You start to see a blip at higher levels in the wild type, and now you see giant currents in these hypersensitive mice. And when you do that knock-in, now you can get place preference to levels of nicotine that are completely ignored. They simply have no effect in a wild type animal, but now they cause a place preference in these hypersensitive animals. And what that gives you is the flip side of, do you need these receptors? Yes, because if you knock them out, you don't get these initial responses to nicotine. Are they sufficient? Yes, because you can go down in levels of nicotine very, very low, and now you can get reward that is mediated presumably through these hypersensitive receptors. The one caveat is that at these very low levels, you should be also engaging, for example, those homomeric receptors that are giving you nicotine-mediated glutamate release. So we don't know if this response, for example, would also occur in the absence of alpha 7. That hasn't been done. It's a real pain in the behind to do, and maybe not worth it. Same is true for alpha 6. You get these very, very large currents in dopamine neurons with this hypersensitive mutation, and then you get hugely increased activity in response to nicotine in mice. And if you use the most powerful mutation, so that L247T rather than some other mutation, you actually kill the dopamine neurons over time. You do get loss of dopamine neurons in these older animals. Then you can do the opposite. You can take the knockout and you can rescue it with a viral vector-mediated expression in particular brain areas, and this Uwe Maskos did in Jean-Pierre Changeux's lab. You knock out beta 2, and then you put beta 2 back in with a lentivirus, and that's the VTA. And what you can do is you can rescue nicotine-mediated dopamine release. There's wild type, knockout, rescue, and you can rescue intra-VTA nicotine self-administration. And that's wild type, knockout, rescue. So it gives us the VTA, it gives us necessary and sufficient, and then we need still the dopamine neurons. The alpha 6 hypersensitive probably gave us the dopamine neurons, but we also had noradrenergic neurons. Here, T.K. Booker and colleagues were able to knock out alpha 4 only from tyrosine hydroxylase expressing neurons, and what they found is that they could, and this is a complicated slide, but basically you can get nicotine place preference only in either the wild-type animals, the animals that have the Cre only, the LOX only, and also you lose it if you get rid of alpha-4 completely, or if you only knock out alpha-4 in the Th-positive neurons. So I'm going to stop hour 1 there. Yeah?
So if you were to pre-expose a mouse for a while nicotine,
Yeah.
the upregulation of
Yeah.
those receptors, would you be able to use this really small doses to get CPP?
So that was done, and there was a paper done in rats that basically did exactly that. They did chronic low-level exposure, got the upregulation, and showed that in a rat strain that did not normally show CPP, they now got CPP. And that was done by, I think it was Mohammed Shoaib and Ian Stoleran's lab. I may be wrong. But we tried to reproduce that with another strain of rats, we're not able to, and we can't do that in mice. But that doesn't mean that doesn't happen in humans. And it may be that you have this interaction between the overall sensitivity, perhaps, of the dopamine system or the other nicotinic receptor subtypes that feed into that system. So it may be very gene dependent, genetic background dependent.
So why would you get escalation of smoking
Right.
over time with that?
So there are two issues there, right? One is that there is not escalation of smoking in every human. So genetic background, I think, plays a tremendous role in human smoking as well as in these animal studies. We can get giant strain differences in the animal studies. We don't do strain studies in humans. We do linkage studies or association studies, and you see the same thing. There are people who must have a sort of a cohort of other genes that allow them to escalate their intake versus those who try it. Maybe they would even be chippers. Maybe they wouldn't want to continue at all. That's answer 1. Answer 2 is that in the studies that we're doing with experimenter-administered nicotine, we're not actually doing any behavioral training during the time that we're administering those low-level nicotine shots, basically. And it probably is the fact that what nicotine does extremely well is to reinforce whatever behavior you're doing when you get it, not only because of the dopamine neurons, but also because of the prefrontal cortical circuits that are also potently modulated by nicotine. And it may be as simple as you really have to have a control over that administration for it to become salient and for it to actually pattern behavior. There's probably some balance between habit formation, not in the trivial commonplace description of habit as it's something that is a bad habit, but in the fact that you switch behaviors from the nucleus accumbens up towards striatum that you just don't get when you give animals shots. Sure. Yeah.
I have a question. You mentioned the L247 or 67T.
Yes.
Is that the homozygous?
Right. No. So in
is it
fact,
heterozygous
most
or
of those
is it-
are heterozygous. When you get to the homozygous state, in fact, you start to get more loss of neurons. One copy of the alpha-7 knock-in, for example, is enough to get the degeneration of cortex and hippocampus. For the alpha-6 studies, what they did was they actually made a number of mice with various knock-ins, and the ones that really worked best were the ones with the least hypersensitivity. You could still go way down in dose, but you probably didn't have choline stimulating the receptor the way that you would.
I'm just wondering, does that happen in a human?
So people have looked for the L247T, have not found that particular mutation. There are now a number of rare polymorphisms that actually do change the coding region of various subunits. One of the great things about deep sequencing is that you can sequence a lot of people and find these mutations. They're so rare that it is really very, very difficult to get some kind of causal link between smoking behavior and these variants. They seem to be, at least in one study, Joel Galernter reported that they were underrepresented amongst some of these rare mutations were underrepresented among smokers. It's really too small an N to make that conclusion. But maybe as we start to get many studies together and there are meta-analyses that can be done, we can answer those questions. We can see consequences of those rare variants on function of the receptor, both in cell lines and in oocytes. So there are human mutations that change the affinity and the desensitization kinetics of these receptors. Not as profoundly as the L247T mutations.
So the human studies with common variants, why do you think it's only alpha 5, alpha 3, beta 4 that have come up with such strong hits, whereas a lot of these other subunits really aren't?
Right.
Strong.
So, I mean, I think there are two answers. One is that the dark side is very, very powerful in promoting ongoing smoking. And so if you have a change in the aversive properties of the receptors, that it has a really big bang in terms of how much nicotine you'll take in and how much exposure you'll have and how much more likely you are to progress to addiction. I think the other answer is that the other subunits are less well localized. So the cluster alpha 3, alpha 5, and beta 4. Alpha 3 and beta 4 are ganglionic. They're in the habenula and interpeduncular nucleus, lower levels elsewhere. Alpha 5, in addition to the habenula and interpeduncular pathways, very highly expressed in deep layers of cortex. And if I get to my data, I will actually show you some of that. Both much more highly localized than these high affinity receptors that are in the VTA. I think that if you had a real null for alpha 4 or beta 2, that you would see a profound effect on smoking behavior. We don't have a real null, and it may be that we can't really have a real null and have a really well-functioning human. The mutations that are powerful that have been identified in alpha 4 and beta 2 cause autosomal dominant nocturnal frontal lobe epilepsies early in development. And those kids have learning problems, they have reading problems. It is, again, rarer than these polymorphisms that may affect function of those receptors less. So it's been, again, difficult to say whether that protects or protects you against or promotes the initiation of smoking. But I do think that what you can find is what natural variation gives you. So these alpha 5 mutations are obviously extremely powerful, maybe partly because you can survive with them, and that they don't actually cause some other deleterious effect that would make them much rarer in the population and therefore harder to study.
It also explains their weaker effect sizes too.
Yeah.
Also,
John?
alpha 3 and beta 6 do come up in
Beta
genes.
4, yeah.
Chromosome.
Yeah. Yep.
Marina?
Yeah.
Do you think that the alpha 5 ASM 398 variant is altering nicotine affinity or channel opening after nicotine?
Right. So some very nice studies have been done by Jerry Stitzel and others. The kinetics of the receptor are greatly modulated by inclusion or exclusion of wild-type alpha 5. It really greatly increases the affinity for nicotine or acetylcholine. What the polymorphism seems to do, at least in the studies that are reproducible, seems to be changing potentially the calcium influx through those receptors. It does seem to affect the open time of the channel. So I do think it is clearly changing the properties of the receptor. What aspect of that change is really essential for its effect on behavior, I think is really completely unknown. Because there are a few.
Can I
Jonathan,
one more?
yeah, go ahead.
Which is, it's fascinating that there's this transitional state where that the nicotine receptors go into.
Yeah.
So what does that mean for addiction to chewing tobacco, where it is being absorbed through the mucosa and it's much slower and more steady, and as if you were really chewing nicotine gum, I guess?
So there are two issues. One is that even in a fully desensitized receptor, there is still what's called, I think Roger Papke calls it the smoldering current. They are not off, they are just greatly decreased. So you will still get nicotine-induced dopamine cell firing. You will still get some dopamine release. It'll be much, much decreased. So it may be that that's enough to begin to drive the processes that lead to chewing tobacco habit addiction. It is not clear that the hallmarks of chewing tobacco addiction are the same as smoked nicotine addiction. And really the best analogy there is the crack cocaine epidemic. There was a lot of discussion about whether cocaine was addictive for many years, just as there was about tobacco. And there was no way to ignore it once people started smoking crack. The kinetics are so different, and the power of the rewarding experience and the reinforcement is so different between those modes of administration that we had crack addiction and we have cocaine abusers. There's no question that there's cocaine addiction from other sources of intake as well, but there's a real quantitative as well as qualitative difference. I think that's also true for snus and chewed tobacco. The same is probably going to be true for tobacco replacement. You can probably get some addiction to tobacco replacement, but is it the same as addiction to smoking? The overall power over behavior is very, very different. Jonathan?
I was going to ask about the role of these various receptors in terms of impulsivity and delayed discounting.
Yeah. So that's actually, let me just quickly show you data that you already saw from Paul Kenny that the alpha-5 knockout increases nicotine self-administration. You didn't talk about it? Okay. He was on a hypocretin roll? Okay. So then let me answer your question first. So what we do know is that human smokers, just like human heroin abusers and human cocaine abusers, in large studies from the person whose lab I cannot remember at the moment, but I can give you the references, do greatly increase the steepness of the delay discounting tasks, delay discounting performance in humans. So delay discounting, I'm sure you've already heard, it is basically the question of whether you will take a smaller reward today, how much of a reward today will you take instead of a big reward later, right? So we all have to do that in order to get to graduate school, through our assistant professorship. We have to delay gratification in order to get through difficult times. It's very important for human behavior. And if you're an abuser of particularly of any drug of abuse, you will actually accept much lower rewards today, even if you're promised greater rewards later. So human smokers definitely have a very steep delay discounting curve. In animals, you can also see effects of chronic nicotine exposure in rats on those delay discounting tasks. But I think what we don't know is whether there is simply, or it's not simple, but whether that's due only to the effects on the dopamine neurons, or whether the fact that there is a great deal of prefrontal cortical, let's say microcircuit activity that's modulated by nicotinic receptors that also would affect, for example, the ability to suppress negative behaviors, the effects on impulsivity. I think nicotine may be worse than some of these other drugs for those, because it is having profound effects outside of the mesolimbic system in addition to those effects on dopamine neurons. So it does do the same stuff. The doses are different, you know, the kinetics are different, but I'm wondering if they're maybe in fact more long-lasting, and maybe not more long-lasting because the effects of other drugs are also long-lasting, but more complex effects of nicotine. So let me quickly show you Paul's data, because he didn't. So here's another slice through the brain, and what you see here, what you see here is a slice through the thalamus, and if I get to my own data, I will talk to you about these thalamic nicotinic receptors. You can see here those widespread high affinity subunits again, and there's alpha-3. Can you sort of see alpha-5 there? Lower level, but it's there. You see this, that deep layer of cortex? It's really only layer 6. Alpha-5 is expressed at much lower levels than some of the other subunits, but really in interesting patterns. And there's beta-4. So this is a cluster that you've been hearing about that is tightly associated with not only the likelihood of smoking, but also now alcohol and lung cancer. And what you can see here, and this is straight from Christy Fowler and Paul Kenny's paper, is that if you knock out alpha-5, and he did this with an escalation protocol where these animals are self-administering nicotine for many hours a day over, and it's very high doses. And what you can see is that if you increase the dose, you get a lot less nicotine self-administration in wild-type mice. Same is true in rats. But if you continue to increase the dose in these knockout mice without alpha-5, they simply plateau and they really don't come down. And if you look at how much they escalate, it's just astounding. You'd never see a wild-type rodent do this. And he then rescued alpha-5 specifically in the medial habenula. And what's cool about that is, so we do have alpha-5 in those deep layers of cortex. You could imagine alpha-5 could be working elsewhere. But when he put the lentivirus back into the medial habenula, what you can see is that it made those knockout mice look exactly like the wild-type mice. He restored the dose dependency, that really tight, pencil-thin dose-response curve that seems to be really a hallmark of smoking and also clearly of nicotine reinforcement in rodents. So it really suggests that the dark side is as important, if not more important, than the bright side. And then there's another piece of data that suggests that this cluster and the beta-4 cluster in particular may contribute. And what she did was she took a BAC that expressed the whole cluster, beta-4, alpha-3, and alpha-5. I think she mutated alpha-3 and alpha-5, so she was mainly putting back in beta-4. And what she could do is to overexpress beta-4, and she would decrease nicotine CPP. And then she rescued it by putting alpha-5 into the medial habenula and upregulating alpha-5. So it suggests that that cluster is really important in the basal activity of the habenular interpenuncular pathway, which we can think of as a pathway that is intimately and reciprocally connected with the mesolimbic system, and that normally acetylcholine is probably regulating the activity of that pathway profoundly. There are a lot of nicotinic receptors there, and that changing the balance of nicotinic receptors in that brain area, and maybe that's what's happening in the folks with the polymorphism in this cluster, or various polymorphisms in this cluster, particularly Mr. Big in alpha-5, is that you are switching the balance between the part of the pathway that really mediates the aversive effects of nicotine, interfering with the normal regulation of acetylcholine, of the balance between those two pathways. So that says that these high affinity receptors that probably contain both, in some populations contain alpha-6, some populations contain alpha-4, some contain both. They contain the beta-2 subunit. They're absolutely essential for that initial step in starting this whole pathway to addiction, which is to open those nicotinic receptors, presumably on the dopamine neurons, and there's pretty good evidence. And that starts off the firing, the plasticity in this system leads to increases in extracellular dopamine levels, and then a bunch of behaviors that we can measure in mice, and we think smoking in humans. However, at the same time, nicotine is binding to these other classes of receptors. They probably contain the alpha-3 and the beta-4 receptor. They certainly contain the alpha-5. There is beta-2 and alpha-4 in this medial habenula interpenuncular pathway as well, just much less. So alpha-5 could combine with both, which is important. Remember, those receptors are also getting activated by smoked nicotine. It is probably normally limiting the activity of the dopamine neurons, and it's limiting the amount of nicotine that you can actually take in. And there are also dopamine neurons that are impinging on, GABA neurons that are impinging on these dopamine neurons that are probably also part of this circuit, and that is also equally important to limiting nicotine self-administration, mediating. It also mediates nicotine withdrawal in animals. So that's where I am with hour one. Good one. Yeah, so I'm going to give you guys a break, and I will give you a really rapid zig through a completely different topic, if that's okay. Unless you have a lot of questions, or unless you don't really want a break. You need a break.
5-minute break?
You definitely need a break.
Okay.
It's good I only plan to tell one story in the second hour. I mean, second hour. I know.
One of the questions.
Yeah. Yeah, oh, should have mentioned that. Actually.